Hello and welcome to the MyHeritage webinar series. I'm Jeff Rasmussen, your host, broadcasting to you live from Middleton, Idaho. Today we get to learn about the science behind MyHeritage DNA testing. Doesn't that sound fascinating? And we will learn straight from the scientists and product managers at MyHeritage. Both Ethel Voll and Gal Zrihen are here with us from MyHeritage headquarters in Tel Aviv, Israel. So nice to have them with us. Also nice to have all of you. Ethel Voll will speak to us first. Ethel is a bioinformatician at MyHeritage. She has an MSc in genetics and bioinformatics from the Hebrew University in Jerusalem. She is a member of the DNA science team, which expands the company's expertise in the realm of DNA analysis. She researches genetic data algorithms to develop and upgrade MyHeritage's ethnicity, matching, and other DNA-based products. Gal Zrihen will then speak to us. Gal is a product manager responsible for MyHeritage DNA products. She leads a talented team of developers, QA engineers, and designers to create and optimize DNA users' entire journey, from ordering a DNA kit to making the most of their DNA results for genealogical research with MyHeritage's constantly growing suite of features. So please put together your virtual hands and let's give our speakers a nice warm webinar welcome. And let's first say hello to Ethel. And Ethel, welcome to the webinar. And Ethel, there you go.
Hi.
Hi.
Hi, Jeff. Thank you.
Yeah.
Really excited to be here.
Oh, it's nice to have you here.
Let me show my screen.
Looks good. It's all yours.
Okay, great. So I'm really excited to be here to talk to you today about the science behind the MyHeritage DNA testing and how do we work with genomics data in order to give you personal insights about your DNA. Our mission at MyHeritage is to enable millions of people to discover their past by finding more about their relatives and about their ethnicities and where do they come from, and empower their future. Today's talk agenda is going to be about, first of all, presenting to you the MyHeritage science team. What do we do, and what our expertise are. We're going to talk a bit about genetics. So at first, we're going to talk about what is DNA. We're all talking about doing DNA tests, but do we understand what it even means? We're going to talk about genetic inheritance. And we're going to use this knowledge to understand how do we do DNA matching at MyHeritage, and how do we predict our family relationships. Then we're going to talk about genetic groups. This is one of our products that allows us to further understand the populations that we are coming from. And then Gal is going to talk about with you about how to get the most out of your DNA features and tools. She's going to give you interesting tips how to identify family lines of your DNA matches. And in the end, she's going to announce a new feature. Okay, so let's begin. As Jeff mentioned, I'm a part of MyHeritage science team. And in the science team, our mission is to take all the data we get from the DNA kits and to extract valuable information from it. For this, we have people who are coming to us from the academia. All the people in the team have second degrees and doctorates. And they have a vast field of knowledge in bioinformatics. Bioinformatics sounds like a big word, but actually this is the combination of biology and computer science. This allows us to take biological data such as DNA and to understand how to do mathematical and statistical tests on it and to understand as much as we can from it. we have people who experts on population genetics. This helps us to understand best how inheritance work, and how can we understand more about the populations that people are coming from. Since DNA contains a lot of data, and we're working with more than 6 million DNA kits, we need the people who understand how to do large-scale computing, how to make our computational work more effective. And we need people who understand machine learning, because we do a lot of predictions about your family relationships and about your ethnicity. So to sum it all up, we're using our scientific knowledge to do DNA analysis and research with cutting-edge technologies in order for you to get personal insights about your DNA. The main products that we develop in our team are, first of all, the matching, which we're going to talk about today. It means that we're taking your DNA and we're comparing it to other DNA kits in our database in order to find your relatives. We're developing the ethnicity estimate and the genetic groups, again, which I'm going to talk more about in the next slides. So let's start with what is DNA. DNA is a very long chain of molecules that contain all the information that is necessary for the life and function of all living organisms. And I like to think about it as the recipe book of life. Here we can see a human. All humans are made of many cells, and each cell contains 46 chromosomes. And when we look closely at these chromosomes, we can see the DNA. DNA is a double helix, which contains nucleotides. We can see it better here. This is how chromosome looks under the microscope. And if we look even closely, we can see that the DNA contains these letters, which we call base pairs. We have 4 types of letters: T, A, G, and C. So the human body is made out of 10 to 100 billions of cells, and each of these cells contains the entire copy of our genome. It means that it contains our entire DNA. And our DNA is made of more than 3 billion base pairs, 3 billion letters. And inside those 3 billion letters, we have about 20,000 genes. And a gene is a DNA unit that carries the information that determines your trait, such as the color of your eyes, the color of your hair, and so on. And if we're continuing with the metaphor about the book, we can look at the genome as a book. And the book is divided into 46 chapters, which called chromosomes. We actually have 23 pairs of chromosomes and about 20,000 genes, which we can treat them as the recipes. These genes contain the code that tell our body how to interpret them into proteins and into our traits. As I mentioned, we have 23 pairs of chromosomes. Chromosomes 1 to 22 are called autosomes, and we get one copy from each parent. You can see here the pink copies we get from our mothers, the blue copies we get from our fathers. And in addition, we have gender chromosomes, which males at birth get X and Y chromosomes, as you see in this picture, and females at birth get X and X chromosomes. So instead of Y, females have 2 X's. The DNA is not something that is unique to humans. All organisms have DNA in all of their cells, and we're actually more similar to many living organisms than we might think. For example, we have 95% of similarity of our DNA to a mouse. We're 94% similar to in our DNA to a dog, and 98% similar in our DNA to a chimpanzee. So if we see that we have so much in common with other organisms, is there a single human genome? Do we all have the same genome, or do we have different genome? So as it turns out, 99.9% of our DNA is similar across all human beings. It means that all people on the planet share 99.9% of their DNA, and only 0.1% of our DNA is different. This difference is actually about 3 million base pairs. It means 3 million letters. But this number is not exact for all human beings. For example, for identical twins, there are no differences at all. All of their letters, their entire genome book is completely similar. But for people who are unrelated, who are not coming from the same family, people who are coming from the same ethnicity have 0.07% of differences in their DNA. And people who are coming from different ethnicities have the maximal differences between humans, which, as I mentioned, it's 0.1%. Okay, so now we know what DNA is, and we want to talk about how do we inherit our DNA from our parents and from our ancestors. And in order to explain it, let me start with an example. We have here two people. We have Dana and we have Eric, and we see that each one of them has 2 chromosomes. Of course, they have 46 chromosomes, but here I want to show only 2 as an example. As you can see, Dana has a purple chromosome she gets from her mother, and a green chromosome she gets from her father, and same for Eric. Then Dana and Eric meet and they have a baby, and each one of them gives the baby David exactly half of David's DNA. You can see here that David has a DNA that is a mix of Dana's 2 chromosomes and a mix of Eric's 2 chromosomes. So David gets exactly the half of DNA from each of his parents, but he doesn't have the exact but because Dana's chromosomes mixed before inheriting them to David, he doesn't have exactly 25% of DNA from each grandfather and grandmother. You can see it here because David has more green DNA that he inherits from his grandfather, Dana's dad, compared to the purple chromosome, which comes from Dana's mother. Similarly, we have here Linda and John, who also each one has 2 chromosomes, one from their father and one from their mother. And then they have a baby called Susan, and she gets exactly 1 chromosome from her mother, Linda, and 1 chromosome from her grandfather, John. And again, Susan has the half of her DNA from her mother and from her father, but from each grandparent, she gets approximately 25%, but not exactly. As you can see here, she has more green DNA than yellow DNA. And then Susan and David have 3 children. And as you can see, again, each parent has a mix of his 2 chromosomes and inherits the baby only 1. So from this, we can understand that, first of all, exactly 50% of our DNA is inherited from each parent. But from each grandparent, we inherit approximately 25%. It means that we can have 20% from one grandma and 30% from other grandpa. And it also means that full siblings generally share around 37 to 50% of DNA, and this percentage can vary. Here, you can see a bigger picture with the values of DNA percentage you share with your family relatives. Again, the percentage that is shared with your parent and child are exactly 50%, but all the rest of the percentages are an average percentage. For example, you share approximately 25% with your grandparent. And as you can see, the farther along we go from the purple dot on this chart, the less DNA you share with these relatives. For example, you share 50% of DNA with your child, 25% with your grandchild, and the more generations it goes, the less DNA you share with your ancestors or descendants. Another interesting fact is that on this tree, we can have a few relatives that we share approximately the same amount of DNA with. For example, we share 25% of DNA with our grandparents, with our uncles and aunts, and with our nephews. And there are ways to separate them when we're doing a DNA test, which we're going to talk a bit more in the next slides. Another interesting point I wanted to talk with you about is that there's a difference between your genealogical ancestors and your genetic ancestors. You can see here in the blue point you, and when we're going one generation back, you have a father and a mother, biological father and mother, which you inherit DNA from. Then we have 4 grandparents, which we also inherit DNA from, and we have 8 great-great-parents, which again, we inherit DNA from all of them. And if we go more generations back, we start seeing here white squares. White squares are people who are our genealogical ancestors. It means that they are the parents of the parents of the parents of our parents, but their DNA got so diluted in this process that we're not sharing any DNA with them. And if we're going even more generations back, you can see that the number of ancestors we have that we don't share any DNA with goes higher. So some of our genealogical ancestors might not be our genetic ancestors. And if we go 11 generations back, we can see that we carry genes from less than half of our direct ancestors from 11 generations back. And another interesting point is that for every DNA position, for every DNA segments we have, we originated from a single ancestor. It means that every segment of DNA, if it's a bit longer, a bit shorter, it comes from only one ancestor. And then when we put together our entire DNA, we have a lot of segments that we get from a lot of direct ancestors. And another interesting fact I wanted to share with you is that all humans have a common genealogical ancestor, who lived around 200,000 years ago. It means if we draw our family tree many generations back and we get 200,000 years ago, we're all going to get to the same common ancestor. Okay, so now we understand inheritance, we understand genetic, what is DNA. How do we do matching in MyHeritage? So let's get back to the example we talked about before. You remember Linda, who got one blue chromosome from her father and one red chromosome from her mother. And let's say she did a DNA test. And when we get her DNA results, we get them mixed. It means that we don't know which letter comes from which chromosome. We have two letters for each position on her chromosome. Of course, if we have the same letter in the same two letters in the same position, it doesn't matter because it means that she inherited in this place C from her mother and from her father. But in case we have two different letters, we don't know exactly how to separate those two parental chromosomes. For this, we have a technique that is called phasing. Phasing is a technique to separate those parental chromosomes, and it allows us to get the whole segments, one from the mother and one from the father. Of course, if Linda's parents did not do a DNA test, we can't say which one gets from the mother and which one she gets from the father, but we do know to separate them in order to compare them to other DNA segments of other people and to help her find her relatives. And now we need to compare those segments to other people. And we do it by measuring IBD segments. IBD is a shortcut for identical by descent. And this means that we're looking for identical segments in two or more individuals if they have inherited them from the same common ancestor. So you can see here, for example, inside the red square, you can see here a father that is represented in a square and a mother that is represented in a circle. And both of them had two children, a boy and a girl. And they inherited their DNA. Next, those two children had their own children. And here inside the red square, you can see first cousins' relatives. And you can see that the orange segment that is highlighted here is an IBD segment. It means that both of these cousins got the orange segment from their grandfather. And this helps us to identify these identical segments and to tell them that they're related. In order to measure the size of this IBD segment, we're using a measurement called centimorgan. And we use it to describe the length of the specific segment of DNA that you share with your relatives. If you go to our website and you review your matches, for example, here you can see Gal's match with a person named Josh. You can see that they share 7.8% of their DNA. And overall, it means that they share 552 centimorgans. They are sharing 28 IBD segments. And the largest IBD segment is around 67 centimorgan long, which means that their estimated relationship is either great uncle or first cousin once removed, or a second cousin. Gal can now go to her chromosome browser and to see those IBD segments. You can see here her genome, her chromosomes, and you can see in purple the IBD segments that Gal shares with Josh. And here on the second chromosome, I highlighted you the largest IBD segment she shares with Josh, which we saw earlier that it's around 60 centimorgan. Okay. And this is helpful to us because we can now separate different relatives using the amount of DNA they share and the amount of the segments that they share. For example, you can see here in the yellow, you can see here, in the red spot, that these are cases of siblings. Siblings usually share a very high number of very high amount of DNA, and also a very high amount of DNA segments. By contrast, you can see, for example, the orange cloud, which describes the relationship of a grandchild and grandparent. They also share a relatively high amount of DNA, but a bit lower than siblings, and they share less number of segments. And these clouds help us understand how to separate between different relatives. Next, after comparing two people's DNA, we need to do it again and again in order to find all of your relatives. So let's say, again, I want to demonstrate it with an example. Let's say we have two people in our database and we need to compare their entire DNA. So we have to do one calculation. But then we have another person who also wants to find his relatives. And she joins MyHeritage. She does a DNA kit, and we need to compare her DNA to the two other people we have in our database. It means that now overall we needed to do 3 calculations. Now we're adding another person to the database, and he also did a DNA kit. And now to find all of his relatives, we need to do overall 6 calculations. And when the 5th person is joining, we need to do overall 10 calculations and so on. So you can see here that the more people we have in our database, the number of calculations that we do goes exponentially higher. So if you're interested in the formula, for n people, we need to do n times n minus 1 divided by 2 calculations. We have in MyHeritage around over 6 million people in our database. It means that we do, in order to find all of their relatives and compare all of the pairs of DNA, we need to do over 18 trillion calculations. For this, we need a very strong calculational ability, and we need to make our algorithm very efficient and very quick. And this is a process that is done very well here. And all of the things we talked about now allows us to see our DNA match list. For example, you can see mine here. You can see here the first 3 relationships I have. I have here my mother, my sister, and my distant cousin. You can see for each one of them the amount of DNA I share with them. For example, I share 50% of my DNA with my mother, which makes sense, and 36% with my sister. Again, as we said, it makes sense. And later on, Gal is going to talk to you about how to extract more information from these matches. But right now, I want to talk to you about further resolutions of your DNA results. We talked about matches. DNA matches allows us to see in the resolution of less than 10 generations. It means that we can find your relatives that are 2 generations far from you, 3, 4, 5, but we can't tell you anything about your matches of more than 10 generations. And in order to do that, we have 2 other products, the Ethnicity Estimate and the genetic groups. And I want to talk more about the genetic groups. The genetic groups is a product that allows you to see if you're coming from a specific population that we identified. And how did we do that? Again, I want to start with a demonstration. Let's say we have an individual here in pink, and we want to look at all of his DNA matches that appear here in blue. And then we want to look at the second degree matches, I mean their matches. And this creates us a web. And then we want to connect all the dots with their DNA matches. As you can see here. You can see here that every 2 people who have a DNA match are connected with a line. And this creates a web. And in this web, we're looking for denser parts. For example, here, You can see here a few people who are all related to each other, all have DNA matches to each other, and we can define them as a family. So in MyHeritage, we don't look at only one person matches. We look at millions of users matches, which creates us a web of billions of matches, which looks like this. And you can see here that we have denser parts and parts that are more sparse. And when we're looking at the denser parts, if we have tens of people, it means these people are a family. But if we have a denser part which contains thousands or even millions of people, it means that they are probably a population. So in our team, what we did, we took 1 million MyHeritage users and their 10.6 billion DNA matches, and it helped us define thousands of genetic clusters. As you can see here, we took the denser clusters, and we wanted to look at each one of them. As you can see here, we took this cluster of matches, but in MyHeritage, we don't only have the DNA matches, but we also have their genealogical data. We have their historical records and their family trees. And this allows us to look at their stories and try to define what unifies them together. So in order to build this data, in order to understand what unifies them together, we use the following information. We used ancestral events such as birth or death, common surnames, migration events. We wanted to see if many people from the same group migrated from one place to another. We wanted to look if the people from the same group share the same ethnicities. And we wanted to look for different groups with the same geographical overlaps in order to look for different groups in the same geographical place. And this helped us to define over 2,000 of genetic groups that helps our users understand better where do they come from in a better resolution than only the ethnicity estimate. For example, again, you can see my results, and you can see here that I have 6 genetic groups. I have 4 ethnicities, most of which is Ashkenazi Jewish, and 6 genetic groups. And if we look in my ethnicity estimate, you can see exactly those groups. And you can learn more about each of your genetic groups. For example, I wanted to further look at my genetic group called Eastern and Central Europe Ashkenazi Jews, who come mostly from Poland, Germany, Ukraine, Lithuania, Belarus, and Russia. Here I can read more about this genetic group. I can read where they are now and where they come from. I can see here how many kits were used when this group was defined. I can see, I can play here with the timeline and see where these people live. For example, I can see here in the yellow spots where these people lived from 1800s to 1850, sorry. And I can see more information here. For example, I can see here the top places they lived, and you can move the timeline and see how the migration is changed over the years. That's it for me. Now I'm going to pass this to Gal, who is going to talk with you about DNA matches, tips, and techniques.
Thank you, Ethel. Thank you very much. Hi, everyone. Good to be here today. So today I'm going to follow up after Ethel showing us the science behind MyHeritage, and I'm going to talk with you about some of my can say my own tips and techniques on how to get the most out of your MyHeritage DNA features and tools, and how to identify your family lines of your DNA matches. So Ethel talked with us about genetic ancestor and genealogical ancestors, and showed us the difference. And we must consider the genealogy and genetics as two tools that we need to combine in order to find the question that we are looking for. So all of us are using the genetics and DNA testing in order to find our relatives and to identify our past, but we also need to consider the genealogical information that we can combine with it in order to get to the exact answer. So let's start with the first steps on how to get the most out of your MyHeritage DNA features. So first and foremost, build your family tree. You would want to build your own tree out far and wide as possible. You want to add ancestor first. You want to go back as far as you can. The more generation you go up in your tree, it will be easier for you to understand the ancestors. Through them, you are connected to your DNA matches. So it's not just enough to find someone that is genetically close to us, but it's the genealogical information that gives us this piece of the puzzle to understand how he's related to us, who is our common ancestor. Now, don't forget your collateral lines. Collateral lines are the lines of the siblings of your direct ancestors, aunts, uncles, great aunts, great uncles, and so on. Now, those are extremely important for DNA matching because at the end of the day, the people that are going to show up in your matches list are generally going to be the descendants of these collateral lines, the younger generation. And now, with these collateral lines, you want to make the tools and features to work for you. You want to get the most out of them. So these tools like theory of family relativity, smart matches, which I'm going to talk about in a bit, shared ancestral surnames that are highlighted for you for each DNA match, they should be helpful for you to understand how you are related, and they use the information from your tree. So the bigger you make it, the more information that you have and your matches. Second, use discoveries. Discoveries make your life easier. It's not just that you should use smart matches and record matches to add more people to your tree, but you can and also should use them to add more information about the ancestor and the people in your trees. Now, trees with higher quality provides more clues to identify common ancestors between DNA matches. The size of the tree, the amount of information in the tree, like birth, death, marriages, migration events, and also the correctness of obviously of the information, these all affect the quality of the tree. Now, third, if the information that you see that there is in the both trees that is used for smart matching is incorrect or was inputted wrong by you or by someone else, MyHeritage cannot see it if it was incorrect. So you eventually you can end up with wrong information that can mislead you. So always make sure that you review the genealogical data in other people's trees and also in your DNA matches, and you use this data only if you trust that it's correct. So you need to review it and you need to trust it. So treat it with caution. Also, afterwards, you can use the DNA results with the genealogical tool. So as I mentioned, the shared surnames, the shared places, the smart matches are all the genealogical tool that completes your research question, how this DNA match is related through me. Last but not least, contact your DNA matches. Be proactive. Sometimes you won't get an answer right away. Sometimes they will answer you after a very long time. But in the end, you both share the same goal, and it's to find more about your family. So be proactive. Don't be afraid. Contact your DNA matches. Share your information. That's the closing final point of the loop to understand how you are related. So these are my first steps. And let's talk now about smart matching and how you can use them. So we said smart matching can help you enhance your family tree. What are smart matches? smart matches are people that are in your family trees, your ancestors or other people that you've put in your family tree, who also match people in the family tree of your DNA match. And MyHeritage found these two people, these two individuals, to be the same person by its algorithms. Now, our algorithms for smart matching are based on information, such as names, dates, places, and other facts that are documented in the family tree. Once we are confident enough that this information indicate that it's the same person, we suggest it to you, and we suggest you to add it to your tree to confirm the smart match and also add information to your tree in just one click. Now, why they are important? Because smart matches can help you determine which family lines and ancestors you and your DNA match share. And I want to show it with an example. So I got my match, and the match name is called Eric, and we share 1.7% of DNA, 122.7 centimorgan. Well, that's not a lot. I don't have a lot of information. And I know it's somewhere between second cousin and second cousin once removed. First of all, I want to confirm it. And second of all, I want to make sure that what is the exact relation. So when I scroll down in my match page, I saw the smart matches section. And as it says, the smart matches are here to increase my confidence in the DNA match. The more smart matches I have between my tree and Eric's tree suggests that there is indeed a genealogical relationship between us. So I saw here Moshe. Moshe is my grandfather. I know it. I've put him in my family tree. And I saw also Eric has Moshe in his family tree. Similar information, similar birth city and years. And we see that Moshe is my grandfather, but for Eric, it's his great uncle. Okay. And we see Eli. Eli is grandfather of Eric, according to his tree on the right. Now I have also Eli, and I'm going to review the information, obviously, by clicking Review Smart Match to be confident that the information is correct. But you can see that Eli, for me, is my great aunt's ex-husband. So what does it mean? So let's look at the tree for now. So I have me in purple, and I know Moshe is my grandfather, right? And I know that Moshe and Eric, the relation between them is that Moshe is his great uncle. So that puts Eric exactly on the second cousin spot. Now, let's look at the other smart match. The other smart match suggests that the grandfather of Eric is Eli. At the same time, Eli should be my great aunt's ex-husband, like my great uncle. So that's two smart matches that confirm the theory that Eric is my second cousin. So I had the genetic data. I had the amount of shared DNA. It suggested me the relationship or the relationship range. The smart matches sealed the deal. They proved me how Eric is related to me and that we share common ancestors, which are the great grandparents of us. So that's one example of how this genealogical information can help us and why smart matches are a crucial part in the research work through the DNA matches. Now let's look at another genealogical tool that can help me learn more about my DNA match, and it's the shared ancestor surnames, which are the surnames of my ancestors, my great-parents, great-great-parents, and so on. And they also play a vital role in the family history research. Now, we identify the surnames that you and your DNA match have in common and bring this to your attention. An ancestor surname that you and your match share could point to the identity of the shared ancestor from which both of you inherited the same DNA segments. So in this example of me and Eric, first of all, Eric doesn't have the same last name as mine. My name is Zrihen as the last name. Eric has a different last name. So I don't have a quick hint here from which side Eric is related to me and how exactly. But once I look at our shared surnames, I can see that Zrihen, which is my last name, points me to the surname of Eric's ancestors. his grandmother, his great-grandfather, and his great-grandmother all share Zrihen. Now, remember, in the last slide, we saw that Eric is my second cousin and is related to me through the most common recent ancestor of the great-grandfather. And their child is Eric's grandmother, which is Alice Zrihen. So that's also an indication, which if I didn't have the information that Eric has Zrihen in his family tree, I wouldn't know that exactly. I wouldn't know from which side. Now I know it's from my father's side. So that's why it's very important to have the information in your trees. Next, we have also the shared ancestral places. Kind of like shared surnames, we look at the information in your tree and in your matches tree, and we see if you have shared places. Shared places refer to either towns, countries, or US states that appear in both trees, and refer to events like birth, death, and migration events. So it's important to look at the shared ancestral places because it's also an indication that you come from the same family. In the 21st century, we have a lot of migration stories that comes from our older generation, from Europe, North America, South America, everywhere. In my case, even Africa. So to know that the DNA match has ancestral places that are similar to mine also helps me to identify from which side we are related and how. Okay, so let's talk about how to identify family lines even more of your DNA matches. And the tips I'm going to give you here are get more data. What does it mean to get more data? If it's possible, test older generations, great parents, parents, uncles. If you're lucky enough and you can test your grandparents, and the higher you can go in the generation, the better it will be. The more matches you will likely cover. So it doesn't mean just great parents. It can also be great uncles. The older generation, the more it's spread through the descendants. If you don't have any older generation to test, then test your cousins. The first cousins will lead you to your grandparents' line. The second cousin will lead you to your great-great-parents' line. That's the rule, and I will demonstrate it in a minute. Now, third thing, use your shared DNA matches. This is the section that will appear in the review match page in every match that you are reviewing. When you review the shared DNA matches, look for the triangulation. Triangulation is the key for you to identify the common ancestor, and if there is any. And I will show it in a second. Now, what I meant by testing more relatives and why it's important to go to all the generations. So going back to Ethel's diagram, in purple, there's myself. And when I go up, these are my ancestors. So let's say I was lucky enough to test it, to get my great-grandparent tested. Now, I share with my great-grandparent 12.5. But if we go to the descendants, other descendants of my great-great-parent, which are or my great-uncles, cousins, once removed, twice removed, now we can see that my great-uncle, which I share with 12.5%, shares with my great-grandparent 50%. My parents' cousin, which is the first cousin once removed, I share with him 6.25%, but with my great-grandparent, he shared 25%. And second cousin share with my great-parent 12.5%. So what do I mean by that? When you get to test older generation, and assuming that you have more relatives that are tested within MyHeritage, and every day the database keeps growing, then once you find DNA matches, these older generation will help you to get to the solution of how they related to you. And let me show you an example. This is me, and I got my DNA match with Gabrielle. Now, at first sight, I'm seeing the DNA match quality, and I'm looking, and I see 1.4% of shared DNA, which is 26 centimorgan. Not a lot, not something that I usually can work with. And the estimated relationship suggests that is somewhere between my third and fourth cousin. Now, this is very far. I mean, these are the relationship ranges that it's hard to identify. Sometimes you say, okay, I don't have anything to do with this information, and I don't have any further need to investigate that. However, I asked my father to get tested, and you can see him in the first row of the shared DNA matches. So after I got my dad results, I've looked at the shared matches between me and Gabriel, and I saw that my dad appeared there. So first of all, I know that Gabriel is related to me through my paternal line, sorry, and that my father, who shares with me 50%, shares with Gabriel 4.4, which is significantly higher than what I share with Gabriel. And look at the estimated relationship. First cousin once removed to second cousin. Now, this is something I can work with. This is narrowing down the results, and I can more easily identify how Gabriel, through more research work, but still very much easier to identify how Gabriel is related to me, or more correct, to my dad, and then identify the common ancestor. So this is an example why it's important to test all the generations, your parents, your great parents, and so on. Now, you may have noticed that on top of the shared matches list, there's this sort button. This is a new feature that we introduced not too long ago. We actually decided to add it after hearing a lot of users asking for it, and we saw how much you love to work with the shared DNA matches and how much information it gives you. So this sorting option basically gives you the option to decide how do you want to sort the shared matches list of your DNA match. You usually get quite a long list of shared matches because after all, we're looking for relatives, and we all have different clusters that we are related through. So by default, the matches are ordered in the way that we take the amount of shared DNA with you, the amount of shared DNA with Gabriel. We sum these both numbers, and then we sorted the matches by the highest amount to the lowest. This is to allow you to see the matches that share the most DNA, either with you or with the DNA match. But now you can also sort this list and sort it by either the shared DNA with you or the shared DNA with the match. So let's choose, for example, the shared DNA with me. So now I see that the first choice is Aaron, which is my dad, and David, which is Gabriel's son and my third to fourth cousin. But if I sort it by the DNA matches that share the most DNA with me, now I see something interesting. Now I have Max, and now I have Daniel here also. Well, Max is kind of far from Gabriel, but look at that. Max and Daniel share a substantial amount of DNA with me, Max especially, above 500 centimorgan. So I can use them to look at them first and not to scroll down through the list. I can use them, see them, identify them. It will be more easy for me to understand how they are related to me, and then from them to move on to Gabriel. So I can switch the view and get information that I need. I can do it by the shared DNA with me, and I can choose by the shared DNA with Gabriel, for example. And if I click that, then we see also matches that we haven't seen before, like Josh, which is Gabriel's half brother or nephew, and Catherine, which is Gabriel's first cousin. And now it's easier for me to take these matches and maybe build Gabriel's tree and then connect it to my tree. So we are going back again to the importance of getting the most information out of your family tree, share it with matches, look at the genealogical information of the matches, and combine the genetic data that you get with your results. Now, if you are not able to test the older generation, you can also test your cousins. So if you test any one of the second cousin, still, it will give you a hint how to go back to your great-grandparent. If you test the first cousin, then it will give you a hint how to go back to your great-parent. So it's the upside down way, but it's still very helpful to identify common ancestors and family lines. So, as I said, and I promised to talk about triangulation and explain what is that. And let's explain what triangulated segments are. So triangulated segments are shared DNA segments that you and two or more DNA matches share with each other, and therefore likely all inherited from a common ancestor. So they call triangulated segments from the word triangulation because we have a party of three or more here. You, a DNA match, and another DNA match. When you all share the same segment at the same location, this segment is called a triangulated segment between all three of you. And we can say that this segment was inherited to you by a common ancestor. And we can link this segment, say, hey, this segment belongs to a common ancestor X. And we have the chromosome browser, and this is an example from the chromosome browser tool, the one-to-many. And I've added here two of my matches, David and Josh. And I see here that we have 7 triangulated segments. So if you look at the image below, it's kind of similar to what Ethel showed before about IBD segments. So now triangulated segments are actually IBD segments that are shared between 3 people and not just 2. So I'm looking at each one of my chromosomes, 1, 2, 3, 4, 5 until 22, the autosomal chromosomes, pair of chromosomes, and the lines. Sorry, the lines you see in red are the segments that I share with David. Now the lines you see in orange or yellow are the lines that I share with Josh. Now you see the bubbles there. The bubbles there mean that we identified the segments that me, David, and Josh all share at the same location, the same information inside these segments, and therefore these segments are all linked to a common ancestor that is common for me and David and Josh. Now, you don't have to do this work manually and go to the chromosome browser and add these matches and see where you triangulate. We also provide it to you automatically. So I'm going back to Gabrielle and I'm looking at the shared DNA matches. And when you go over this this list, and remember, triangulation is a party of 3. So I have me, myself, Gabrielle, and another shared match between the 2 of us. And when I'm looking at the shared matches, it provides me the information if we have a triangulated segment. So in this case, I see that myself, Gabrielle, and David all have 4 triangulated segments, and these segments are the ones that we all inherited from the same ancestor. You still can have shared matches with another match that you don't have any triangulated segments. But if you see this icon, click on it. It will send you back to the chromosome browser, and we automatically identify this information for you so you can see it. So use it. It's a very important tool. So that's it, guys. That were my tips and techniques on how to use genealogical information and tools that will set you up to success in order to find what you are looking for in your research questions. And I want to end my part with a happy announcement on a new feature we launched yesterday, which is the MyHeritage Global Name Translation Technology for DNA matches. Now, you know that one of the strengths of MyHeritage platform is that it's translated into 42 different languages, and that it's the most popular genealogy platform in most of the non-English speaking countries. Also, in addition to its popularity in the English speaking world. And we have billions of historical records and family trees from around the world. So we always try to develop technologies that overcoming the language barriers. And language can often serve as an obstacle when you explore your DNA matches from a different country. So this is a new application of the existing global name translation technology. And now we brought it into the DNA matches. And this is great news for all the users who receive DNA matches in either Russian, Bulgarian, Ukrainian, Hebrew, or Greek. And their names are written in these languages. Or any users that their display language on the site uses Greek, Hebrew, Ukrainian, Russian, or Bulgarian. So now you will find the names of the DNA matches transliterated. Yes, transliterated, because what we do is we're not translating the name, we are converting the name. For example, like here, that I get a match in Russian, which I don't know how to read, but through the transliteration, the translation technology, I can know that this name means Sergey, and it appears to me in different places on the match card. So I have it here in the match name, and in this case, the DNA kit of Sergey is managed by someone else, and I can see the DNA managed by, and I can also identify it's Santino, and now I can contact either Sergey or Santino with the right name, and continue my research with them. So this is a new feature you will see now in your matches list if you have matches that are written in the alphabetical letters that are not according to your display. So we either translate them from Cyrillic to Hebrew and Greek to Latin, or vice versa. And you will be able to see them on your matches card or in the review match page. And also when you get your weekly emails about new matches that we find for you, you will also be able to view this translation there. So I hope it will help all of our users that are getting matches from all around the world in understanding other names. And that's it for me. I want to thank you all for me and in Ethel's behalf. And with that, I will conclude our session. And I invite you that if you have any questions, you want to learn more, you can go into our education center, or you can contact our support teams, and we'll be happy to help you. So thank you all.
And thank you. Congratulations, Gal, on that new feature that was just released yesterday. I love that global name translation, and now that it's available as part of the DNA results, that's a great advancement. You know, it's both fascinating and comforting to have learned about and learned from the science team at MyHeritage. This was very enjoyable. Tracy, thank you for writing this. Tracy wrote, great explanation. I have a much better understanding of DNA now. And Tracy says, I am new to DNA. So welcome, Tracy. Glad that you're here and glad that you found this enjoyable. And thanks to all the rest of you who are writing in with your kind comments. We look forward to this webinar every year. The next MyHeritage specific webinar will be by Daniel Horowitz in about a month from now, December 13th, looking back and peeking ahead 2022 at MyHeritage. I always really like that what's coming part of Daniel's webinar, so be sure to look for this on familytreewebinars.com and register for it. And now let's I'd better go put this link up there for you. So when you're at the familytreewebinars.com, if you just type in MyHeritage webinars, and I'm going to put this link in your chat, that will take you right to the great collection of all of the webinars that are specific to MyHeritage. I put several links in your chat earlier as well to that address in, well, as full-length webinars, some more on some of the topics that both Ethel and Gal were discussing today. So click on that and browse. I'm going to just put another one in there for you as well. So check out both of those links right there. Okay, so let's do some door prizes now. First door prize. So if you're here, you're automatically eligible to win. There's nothing special that you need to do. First door prize will be a 1-year MyHeritage complete plan, and this gives you access to everything on the site. The tree builder, the data subscription, all of the really cool photo tools, all of the DNA tools, and so on. So if your name happens to be Heidi Hanks, watch for an email from me and we'll get that going for you. So congratulations to you, Heidi. And then let's do a MyHeritage DNA test kit. Good for yourself or good for someone else around the holiday times. And let's go and find our winner. It looks like John Hazelwood is our winner, so congrats to you. And there's my questions screen. So thanks everyone for being here, and thanks for sharing some of your questions. So I'm going to just kind of go down the list here and ask your questions of our presenters. So Ethel and Gal, there's a question from Alicia who says, when you use ancestral events like the birth and the deaths that you were talking about, when you use those in the ethnicity estimates, are you relying on people's genealogical trees? And if so, what could you say about checking the accuracy of those trees, or how does that fit in?
Thank you for your question. So actually, the ancestor places that appear on the ethnicity map in the ethnicity estimate are the events that are only relevant to your tree, not the tree of your matches. The ethnicity estimate is something that is personal to you, and you can actually see based on the polygons of the ethnicity estimate results, the same events that you entered in the tree. So you can actually, it's another tool to combine the genealogical and genetics data that you get. There's another feature called shared ethnicities, where you can compare the ethnicities that you got and the ethnicities that your DNA match got, and then also conclude the side or the family lines that you both share. But on the map in the ethnicity estimate are solely the events from your family tree.
Okay. Thank you, Gal, for your answer. Here's an interesting question from Stephen. He says, does MyHeritage have plans to let us override the estimated relationship range with the actual relationship for those matches that we were able to confirm the actual relationships? So I guess, is that possible? And if not, maybe an idea for the future. Any thoughts there for Stephen?
Well, Stephen, that's a very good idea. I can tell you that yes, it's in our plans. We know that it's something that is needed.
Oh, good.
Keep in mind that when you order kits, for example, for your relatives, cousins, or parents, and when you activate the kit from your account and you link the kit, to the individual in your tree, you will have this indication. You will have this indication on the tree with the purple icon, and you will also be able to see it from the matches list. So yes, this is a very good suggestion what you said, and we are it's part of our plans. And besides other cool stuff that we are planning, and very advanced algorithm. But you can also use the icons on your tree for that. Yeah.
Okay, thanks so much. That's exciting. Ernst asks, how small of a triangulated segment can you trust to be IBD?
So I think the question is trust for what purpose? If an IBD segment is longer, it means that it probably belongs to someone closer to you in your family tree. If the IBD segment is short, so maybe it's someone who is farther away from you in your family tree. But of course, that all the IBD segments that we show on our website are IBD segments that we checked and confirmed.
Okay. Is there a threshold of a certain number of centimorgans that is displayed, or if it shows a very small segment, are those filtered out, or can those really small segments be trusted as much?
Well, I can answer that, Jeff.
Okay.
We have some sort of a filtering process in order to provide you the results with the most accurate DNA matches. But if you look at in the slide that I showed about the chromosome browser, the one-to-many, I don't know if you can see my screen again.
I'd be happy to switch over, Gal. Here you go.
Okay.
Thank you. Yeah, looks good.
Okay, great. So if you see here, you have this drop down and it says show triangulate segments that are at least. So you can actually select from, you can select the threshold that you want to see. 2 centimorgan are the minimum that by a rule of thumb considered to be reliable. And you can change that threshold according to the confidence that you want. Obviously, the larger the centimorgans, the more confident you will have. So 2 is the minimum, and you can go up by selecting from here.
Wonderful. Yeah, thanks. Yeah. That's something that I've missed there, but yeah, it's certainly right there. So thank you for clarifying about that. Let's go to Linda. She says, I have a small hand-entered tree on MyHeritage. She's wondering, could I delete that, and could I upload a full GEDCOM file that she has?
So the short answer is yes, you can upload your GEDCOM file into MyHeritage. You should have it under a family tree in your website in the drop-down. And you can select to import a file and get it from other locations if you've built it in somewhere else. So yes, you can do that.
Yeah, very good. Thank you. And then kind of a related question. Helen says that her cousin has their DNA results, but it's in a different service. Is it possible for her to utilize her cousin's DNA in MyHeritage? So can she import the DNA results from a different service into MyHeritage?
Yeah, so we actually support raw DNA files that you can download from either 23andMe, Ancestry, or Family Tree DNA, and you can upload it to MyHeritage. Once you upload, you can pay either one-time fee to unlock all the DNA features and ethnicity estimate. If you don't do that, then you will receive DNA matches. But if you want to get access to all the rest of the features that we discussed today, you can either do it with a one-time fee per kit or a subscription. But the upload itself is free, and you can just download it from another site. If you're not sure how to download it, then when you can Google upload a DNA file to MyHeritage, and we have actually instructions there about how to download files from the other sites, so you can use that.
Yeah, very good. Thank you. Alicia asks, does MyHeritage adjust relationship estimates or filter out known pile-up regions for endogamous populations? And if so, how? That's a big question.
Yes, so of course there are populations that share more similarities to each other than others, and this is taken into consideration when we present to you your relationships For example, we know that Ashkenazi Jews share more similarities than many other populations, and this is for sure something that is taken under consideration and is adjusted in our algorithm.
Okay. Thank you. You're doing very well with all of these questions. Just a couple of more. Annette, and maybe this is a science question. Annette says, if you've ever had a blood transfusion, does your DNA profile change?
Okay, so when you are doing your DNA test, you're taking a sample from a cheek swab. And if you ever received a blood donation, of course, other DNA went inside your body, but it's not something that is going to be shown on your sample in MyHeritage.
Ah, okay. If you were to take a blood sample, I guess that would be different.
I'm actually not sure for how long it's still effective, but it's something very interesting to check out.
Yeah, very interesting. Well, thank you so much. So Alice has a question about her tree. So Gal, you mentioned as one of your tips to make sure that you grow and expand your tree. So Alice is wondering if she uploads a new tree, or if she replaces her tree, does that disconnect the DNA matches in any way?
Wow, that's a very good question. I can say 99% certainty that if you upload a new tree, then it will saved as a new family site, and the DNA kit is actually connected to an individual in your family site. So it will probably lead to a disconnection. But if you do that, you should have the option under Manage DNA Kits to reassign the kit. If for some reason you are not able to do that, you can always contact our support team, but it's something that you can do from the Manage DNA Kits page.
Yeah, okay. Thank you so much. So many of you have asked, will there be a way to watch the replay of today's live webinar? Yes, just head up to familytreewebinars.com, and in about an hour from now, I'll have the replay available for you there. So just click on it, click the play button, and you'll be able to pause or jump around or make the speed go faster or slower, but that will be online, and it will be in the free area within about an hour after we conclude. Ethel and Gal, thank you very much. I know it's late evening where you are in Tel Aviv, and so thank you for sharing the end of your day with all of us. We're very thankful. I forgot to mention at the beginning, we have over 1,100 of you who have registered for the webinar, so thanks to all of you. Any, Ethel or Gal, any final thoughts you want to say before we do say goodbye?
Yes, so first of all, thank you so much for inviting us, Jeff. It was very nice to share our information with you. And thank you all for listening and asking questions and show interest in what we had to say. It was a pleasure.
Thank you, Ethel. It was indeed a pleasure. Gal, thank you. Have a good night there. And for the rest of you, wherever and whenever you are around the world, thanks for sharing part of your day with us. And remember, life is short. Do genealogy first. Bye, everyone.